So this last session of our meeting, it's a great pleasure to introduce Roger BROCKETT from Harvard. And I won't repeat his title, which is up there better than I can say it. Thanks a lot, Roger.
Thank you. And good afternoon. It's my first time at City University, and I'm happy to be here. So today, this talk is being given by neither a biologist nor a computer scientist, but I will try to speak everybody's language to some extent. So what I want to do is to try to introduce you to a point of view and give you something to think about if you're engaged somehow in the subject of mathematical modeling of biological processes. But I will not say anything of relevance to bioinformatics except after one slide. So, point that I want to make is that a lot of processes that we care about occur in stages, and that the fact that many of our favorite modeling prototypes don't admit very well a description of those things, and I'm going to tell you how to make it work in that context. So, what people do, and it has been done in mathematical biology since it existed, was people talk about homeostasis. That is, how do we regulate everything that we need to regulate, from blood pressure to oxygen level in our blood and so forth. There's also been a lot of work on oscillations, breathing, walking, pumping blood, and so forth. However, if you read the biological literature at a certain level, you see that when biologists describe their subject, they often describe it in terms of stages. This happens, then this happens, then this happens. And that kind of thinking doesn't mesh very well with these differential equation models that have been talked about a lot in the mathematical biology literature. So what I want to talk about is how do you make differential equations work for these kinds of problems that biologists would like to talk about or do talk about in terms of stages, and therefore bring us closer to answering the questions in a way that biologists would like to hear the answers. So, as a starting point, you probably all have seen this picture before or something equivalent to it. So the popular picture of cell division, you identify stages. G1 stage, where mostly metabolic. S phase, where it makes it sound like it's mostly mechanical, that is geometric, where the DNA separates and makes two copies, and more metabolic changes. And finally, the physical or geometric division of the DNA, and finally the division of the cell itself. But this is the language that biologists seem to prefer. I was involved, or got involved, with a group at the University of Michigan who was interested in trying to make a model for cell division that would help to refine what something known as cellular reprogramming. And the basic ground rules for this were to take first of all, the basic ground rules were to do everything based on data. So a collection of cells was set aside, initialized by starting them, and then letting them evolve over time. The data that was collected was something, RNA-seq data, which you can think of roughly as being related to protein content of the very large number of proteins involved. These were clustered into something like 2,300 baskets and measured every 8 hours over the cell cycle. Of course, one can immediately think of ways that you would like to have more data, sampling faster, measuring more than just protein content, but also measuring what you can see under a microscope at the same time, et cetera, et cetera, et cetera. But anyway, the goal was to get information about the cell cycle. I want to just mention here one of my complaints about the biological literature, and that is what they call a cycle isn't really a cycle in the sense that Newton would have called something a cycle. And I just put this picture up there to say, look, solar systems go around in circles like this. Cell cycle doesn't do that. It goes around and divides. So to call this a cycle, I think, is a misuse of the English language. Anyway, I'll come back to that. So, in more detail, what did we do with these half dozen or so samples of 2,300 averages? Well, we built a model of this form, a linear model. The X's are these baskets of proteins organized according to something called domains that are naturally topologically associated domains associated with the DNA, and that those domains are determined experimentally, but I'm not going to talk about that. But so we have these this very large vector of data, and we want to put it into a model of this kind. So you say, well, what on earth do you mean? Where's this term? What's it about? All right, so there are special biologically relevant proteins by the name of transcription factors, and it is known that they are important in steering the protein content over the course of the cell cycle. And so this term here is going to be determined later, but what I want to focus on right now is this model here. So what it says is that I know xk, I know xk plus 1. These are very large vectors. A is therefore has a number of unknowns which are the square of the number of components in X. So we would like to determine A from our knowledge of the measurements, but of course it's going to be very underdetermined. There's just not enough data to determine A. We need a principle to determine A. So a good principle, if you're converting measurements into model, is that you shouldn't attribute to the model anything that you can't measure. So what we did in this case was to assume that goodness gracious. Assume that everything else being equal, A would be the identity and nothing would change. On the other hand, we're going to force the condition that A should, for example, A1 should work on Z0 in such ways to produce Z8, but aside from that, it should be as close to the identity as possible. So that was the principle, and that does determine A uniquely, and that gave us the model. Now, what we're going to want to use this model for is to determine what choices of these transcription factors would best dissuade A from going when it divides into two copies of itself, but rather maybe go into one copy of itself and one copy to something that we like. For example, suppose we have skin cells and we would like to have muscle cells. We're going to try to choose U in such a way as to steer a skin cell into a muscle cell. All right, so just put the picture in your mind is we have data in a 2300 dimensional space, and we're going to write down this linear model that hits the data exactly or counts for the data exactly, but which we have no real reason to imagine that the model is linear. And in fact, it can't be. All right. So a little bit of further biology. And I apologize if I'm telling you things you already know. I feel justified in the sense that I've been told that the audience is always mixed, and I'm going to take advantage of that to hide my ignorance. So, not all types of cells can divide, but skin cells have to divide because if you cut yourself, you need to generate some more skin cells as quickly as possible. So they are able to divide, and the same is true for the cells that line your esophagus, et cetera, et cetera. So there's lots of cells that do divide. Muscle cells do not. They are permanently differentiated. It was a big surprise in the 80s when it was discovered that there's a transcription factor by the name of MyoD that would convert a skin cell into a muscle cell with some low chance, but a large, it happened often enough so that it could be said that there was an experiment that converted a skin cell into a muscle cell. And this was done with the transcription factor MyoD. So what we wanted to do is figure out something about the timing and something about the right mixture of transcription factors. Most of the transcription studies that have been done
involve more than one transcription factor. And the cell cycle, which you can think of as being something like a 24-hour period, leaves open the question of, if I have a set of potential transcription factors, and there are, say, maybe 200 of those, and you say, which subset of that 200 should I use, and when should I put it into the cell? Should I put it all in at the start? Would it be better to put it in when the cell DNA is dividing? Or should I save it over here when the actual geometric division happens? Or what? Well, so there is a theory that is based on, I'm going to go backwards here for just a second. Whoops, no, back here to this model. So there is a theory born out of engineering that tells you, if you have an equation like this and you want to convert x from one value to another, what is the least amount of u in a certain norm sense that will carry out that change? And so we applied that theory to our problem, and in that way came up with a very large list of bests, quote unquote, that would convert skin cells into liver cells, or skin cells into muscle cells, or big long list, big matrix in fact, of converting whatever you've got to whatever you want. This is, of course, an imperfect theory because the model isn't perfect. And there's another aspect of this that is special to the cellular reprogramming story, namely that once you put a transcription factor into the cell, you can't take it out. We don't know how to do that. So that means that this U here that we were talking about is subject to a few more constraints than I've talked about. But anyway, we took that into account when we built this table. Okay. So when to put them in is also a question that we can answer using mathematics. So just a little sideline in case you're not familiar with this kind of thing, is that transcription factors are named in the most haphazard way that I can imagine. And so they have cute little names like socks and so forth. So MyOD is another name. There's another one that shows up in socks and MyOD and Oct, etc., are popular and effective in cell reprogramming. And so the question is, where in the cycle do you want to put them in? Now, I also use this slide to discuss one of the points I'll be making repeatedly, namely, is that this staged biological model is as far as you can imagine from my xk plus 1 equals ak xk model, because there's nothing like it. The xk equals this linear function of past is based on a uniform sampling, 8 hours. These segments here are far from uniform. This M phase, where the geometrical division occurs, is much, much shorter than S phase, say. So the linear model is just not very compatible. It's not very easy to talk about that in the same breath that you're talking about the biology description. Okay, so here's something that moves us closer together. In the early 80s, a man by the name of Tim Hunt discovered that there was a protein that he had separated out and measured it as a function of time over the cell cycle, and it followed this red curve here. And he was a bicycle enthusiast, and he called it cyclin. And later on, after some further refinements, people discovered related proteins, cyclin E, cyclin A, cyclin B, so forth, which repeatedly over the cell cycle generated swells and decay, as in this picture here. So this is interesting and mathematical in a certain way, and it carries us a little bit closer to biological description, because you can read off here cyclin E is good in S phase, and although cyclin A is spread out a bit more, it's still these curves relate to the stage description that seems to make the most sense to biologists. All right, there's another. I'm trying to put together here a story which at the end you'll say, oh, I love that story. But I have to put together some pieces. And now a new piece. The new piece is that there are in the cell cycle certain points called checkpoints. And this was discovered by, well, the Nobel Prize for discovering checkpoints, where it went partly to Tim Hunt and partly to another fellow who I'll mention in a minute or two further. So why do we have checkpoints? Well, look, these processes are extremely complicated. How would you like to pull apart the huge number of base pairs required to make up your DNA, replicate them, and put them together again without making any mistakes? Well, you probably won't, sometimes. So you want to check before you go ahead with cell division and produce a monster. You want to make sure that the cell has done the job up until now correctly. So we have several checkpoints that have been identified, and the point of it is if you come along to a checkpoint and it's not correct, the process gets suspended. And there's various ways that DNA, various mechanisms the cell has for repairing DNA if it's made a mistake, but those take time. And so you need to suspend the dynamics of the cell to give it time to repair. All right. I'm going to let go of cells just for a couple slides here, and to say that what the general principles I'm talking about would seem to apply to a number of other things. For example, you have your sleep cycles, and these are things that are evolving according to stages, and this is not science fiction. This is really well understood. And also the model for action potentials, the way nerve conduction occurs, the form in which nerve signaling occurs. And I want to make a point out of the fact that the time between these action potentials varies widely. And much of the information that is contained in this sequence of cycles is contained in the precise differences between the peaks of these cycles. Just to emphasize the non-smoothness of this, if you plot that, if you plot this, think of this as being x, and then plot x versus its derivative, time derivative, and get what we ordinarily think of as phase spring plot, you see something that's really quite unsmooth at the origin, and it sort of comes to this rest point, and then starts again under a suitable incentive. All right, well, this list could go on and on. At the level of metabolism, you got Krebs cycle, a whole list of other things. Oh, I did want to mention that it also occurs at the level of developmental biology, where an embryo develops. That's also a highly staged process. Okay, now I want to talk about a few ideas about dynamical systems. So, one of the things that makes biology a little bit different than some other subjects is the complexity combined with the robustness. So, the fact that we can do arithmetic without regard to what we had for breakfast this morning, and a whole bunch of other variables, the temperature, et cetera, is the tip of the iceberg about how robust biological processes are, and how robust they have to be, because our lives are just spread out all over the place, and we need to acknowledge that. So when you look at a differential equation, it's not like you are only care about a certain initial condition. You have to care about a whole raft of things that are just about the same as this other thing, but they still have to lead to the same conclusion. So that means you shouldn't be talking about solutions as much as you should be talking about the phase portrait of this. That is, not just what happens to one trajectory, but what happens to all the neighbors that go along with it. So that is often approached by thinking of the equilibrium points. I've shown here two-dimensional, but now imagine 2,000-dimensional system. There would still be equilibrium points. There would still be trajectories that are leaving them if they were unstable, and if the cell division process is going on, for example, you want to have some trajectories that leave, you want others to come in. So most of these equilibrium points, or you think perhaps checkpoints, most of these would be of what's called the hyperbolic type. That is, there would be some directions that came in, some directions that came out. And in a little bit more elaborate theory, we would want to know about mechanisms such as the checking mechanism as to how we can control the behavior in the neighborhood of a hyperbolic point like that. So, what are some possibilities for segmented flows? That is, flows that go along smoothly for a while, come up to an equilibrium point, stop, give the system a chance to think a little bit, and then move on. So here are some possibilities. You could take a equilibrium point, you could go out and you can come back to it again. Those are called homoclinic orbits. You could take a equilibrium point and connect it to another equilibrium point. I put this quote in green here to indicate that this is the sort of a model that might come to mind if you were describing the transition between the G1 phase of cell division and the S phase. You would like it to be able to hold up here while you go through the process necessary for checking to see if it's really in good enough condition to go ahead. And then under some other circumstances, you might build a cycle out of these things. I won't talk much about that. Okay, I want to show you something here that I love this piece. Let me show it to you and tell you why it's so marvelous. Do you like that?
Yeah.
Yeah. Do you want to know what's going on there?
Yeah.
Okay. This little 13-dimensional system has 5,040 equilibrium points. And it's only dimension 13, but the best part is that the right-hand side of the differential equation is just linear and quadratic. Nothing difficult. No hyperbolic tangents, no Gudermundians of x or anything like that. It's just linear and quadratic. It's so good that I'm going to show it again. Ah. Yeah. There it is. Okay, so every time this makes an abrupt change, it has come close to a hyperbolic point, and it moves very quickly past there from an incoming stable trajectory to an outgoing trajectory, and it does that, but in this case, a number of times. Now, to give you a way to think about this, is that equation that I showed there, for computer scientists may appreciate this, it's actually, you can think of it as a differential equation version of bubble sort. So what does that mean? You compare neighboring in a list. In fact, the list I had there were the numbers 1, 2, 3, 4, 5, 6, 7. And you have a certain amount of swap space, which gives you another set of 6 differential equations. And then these go back and forth, and they will take whatever list of initial conditions you have and rearrange it so that x1 is the biggest, x2 is the next biggest, and so forth and so on. So that means that you can think about this as going, as being part of a whole lot of equilibrium. I'm drawing it here as if there were just three things to be sorted. So this comes up and sorts 3, 2, 1 into 3, 1, 2, et cetera, et cetera. But there's many paths to the top. And in fact, this flow is in some sense the nicest kind of flow. It's a gradient flow. And therefore, there's something about it, this monotone increasing. And I've given it a name. And I won't tell you what the function is, this monotone decrease, and I've listed its values there on the side. So, just point being that there are simple differential equations that exhibit these kind of properties that would look like staged processes. Okay, but you ask yourself, my goodness, I don't want anything happening in my body that is too delicate. I don't want something in there that if somebody looks at me cross-eyed will ruin my day. So what you would like is that everything that happens should happen for an open set of things around that happening. So I've said here, all right, fine, I'm not going to look at a linear model for these equilibrium points. Instead, I'm going to look for the simplest nonlinear model that has the property I want, namely that it is robust. So what kind of robustness do I want or get? See, in this region, everything ends up more or less the same place. In this region, everything ends up more or less the same place. The red lines are not robust, but you can't have everything. There are plenty of ways because, let's say, mole fractions can't be negative or something like that. There are plenty of ways to make a physical or chemical or electrical system that avoids those red lines and gives you the robust properties that you like. And over here I've pictured what happens is how you can link together these as we did with the example that sorted the list of numbers, but link together things that are robust. Now I should tell you that for the sorting that I showed you just one initial condition. I showed you one trajectory from one initial condition. And if in fact I go back here and I change the initial condition a little bit, I will traverse these equilibrium points possibly in a different order. So this is not robust, but the system, and I don't know how to do that with robust things at this point, I haven't tried, but here's a setup that is robust. And furthermore, since we're thinking about this checkpoint business, so you say, all right, you just equal. Okay. Suppose this were your description, and you came along and the system tells you, uh-oh, we didn't do the DNA separation correctly. We've got a single strand break somewhere. We've got to stop and give the system time to fix that break. How can we control the evolution of this system? Well, one thing you can do is just add a control that looks like this. U times this skew symmetric matrix that will rotate X and Y. So if you turn U on, you'll take something that might have thought it was going to go on to the next stage, rotate it into this segment, and then it will come back and rest there until that job gets done. So we're not only interested in getting autonomous models that would be like an open loop or unregulated playing out of this game, like a record player. But we also want to build into our understanding of this the capability of stopping and correcting if we need to. All right. So, in this way, one can imagine of building a sequence of building a model with a sequence of equilibrium points and trajectories that join these in a robust way, come up to an equilibrium point, and if there's no impediment to going further, just follow the natural flow from the hyperbolic trajectory and then move on to the next stage. So, in some kind of pictorial terms, we would be picking out, in the sorting example, we would be picking out a particular trajectory that we would want to make robust, and there would be a way to make it robust using that U terms I talked about before. Okay, one thing that we should acknowledge is that if you think of, let's say, the heart or the mechanism of breathing, clearly there are stages in breathing, and we want those to be robust, but they are, in the main, you can think of them as being periodic. So this story about embedding stages or describing stages that might happen for breathing, for example, you might want to use a model like that and think of it as having been embedded in a periodic solution. So that's a possibility as well. And there's easy come up examples of why you might want to do that. Okay, just to be a little bit more specific about that, those equations that generated some of the examples earlier, here's what they look like. They're not only are the right-hand sides simple functions, but it's also true that the connectivity of the differential equations is extremely sparse. It's about as sparse as it can be. It's nearest neighbor coupling. X1 couples to Y1, and Y1 couples to its neighbors, X2 and X1, and so on down. So you have just a list of things with nearest neighbor coupling, and then a rather simple looking right-hand side. You can even rewrite these equations in such a way as half of them are linear, but I won't belabor that point. All right, so remember the graph I showed from about cycling and the timing of the cell division process? Well, here is a trajectory generated by these 5 equations. Looks like it could be the kind of thing that a model for the cyclone would need. And again, if you put it in phase space representation, you see how even though these equations are very smooth looking, the trajectory in phase coordinates looks very distant, unsmooth. And there's a 20 degree of freedom periodic solution made up of equations of that kind. All right, so we need sometimes to explain what we see, we need temporal patterns. And we need things to be sequenced correctly. A before B, if we don't get A before B, the cell's going to die, or something of that nature. These things are not limited to the kind of protein replication and so forth that go along with cell division, because cell division also depends on mechanical things, like the mechanical stress that come from the neighboring cells. And there's nothing about these equations that is wedded to any particular kind of technology. All right, so I've said pretty much what I wanted to say, and Professor Smale told me it was okay to stop early, so I'm going to do that. But I do not want to leave you with just some idea that this was a lot of fuzziness. Okay, linear models are the first thing to try, always. I mean, anybody would be crazy if they didn't see if a linear model works. But qualitative features often just don't go along with linearity. So don't cry about it, do something about it. A number of biological processes, including cell division, but not by any means just cell division, proceed in stages. And still, we would like to describe them the way most things are described in the mathematical physics world, by differential equations. But they will require nonlinear models. We illustrated some concrete types of equations whose solutions evolve in stages. So we know it's going to be, it's sometimes possible to describe these things with ordinary differential equations. And we gave a prototype control model that those equations that had the U in it for the control of the flow around hyperbolic points. Thank you for your attention.
So we certainly have time for questions. Questions?
I begin first.
Okay?
So Professor, so could I consider this kind of modeling system as a general equation for describing the conditions? Could I consider it in that sense? So that if there is like an environmental changes that might affect the biological cell division system, so I just add some correcting terms, could I do that in the current stage of this kind of modeling?
If I understand your question, you want to say, how do I put into the equations the feedback that will either let it go forward or not go forward? Okay?
Yes.
So, to do that, you would have to postulate a measurement process. So, I haven't taken on that job of measurement process, and frankly, I'm not sure how. I know DNA, I'm told that DNA can repair itself. I'm not sure what the measurement gives you. Okay, DNA has a double-strand break, say. How does the system know that? I can't tell you, but I feel confident that it must produce some kind of a change that can be described in terms of things other than magic. So I can then use that number, whatever it turns out to be, to apply it to U, convert it into a U signal, which will then say, stop. I'm not going to go any further for now, and you guys go off and do your thing, and when you've got it right, go back and tell me. I don't want to model the measurement process at this point.
But it could be like described in the model as well, right? This kind of measuring, like predicts some kind of unknown measurement phenomena.
I assume so, but, and that's my ignorance, I'll bet you get out even Wikipedia, you can find out what signal is generated when a DNA is not correctly presented.
So,
Yeah.
other questions? Other people have questions?